All right, today we're diving into some really fascinating research. It's all about a clever new way to fight a super common cancer, not with more brute force, but with a whole lot more strategy.
So let's just jump right in with a big question, the one that's at the heart of this entire study. We've got a powerful, widely used cancer drug that, well, it's often failing. So what if the answer isn't a stronger drug, but a smarter one that literally cuts off the cancer's fuel supply? Okay, first things first, let's set the stage. We're talking about a very common enemy, colorectal cancer, and this huge hurdle that doctors and patients are up against all the time, drug resistance. The stakes here couldn't be higher. Colorectal cancer isn't some rare disease. It's one of the most prevalent and dangerous cancers out there for adults, and that's exactly why finding new ways to treat it is so urgent. And listen, this problem isn't just sitting still. It's getting worse. Get this, some regional data shows that cases have shot up by a staggering 90% just since 2015. That's a huge jump, and it really tells you just how urgently we need new, innovative solutions, and we need them fast. So here's the core of the problem. Doctors have this powerful tool in their arsenal called oxaliplatin. It's a standard chemotherapy drug. The trouble is, over time, cancer cells, well, they get smart. They adapt, they learn to survive the treatment, and it just becomes less and less effective. And that is the exact problem these researchers wanted to solve. So that brings us to their new idea, this really cool hypothesis. Instead of just trying to hit the cancer cells even harder, they started asking, what if we could weaken them first? What if we could just starve them? To really get this, you've got to know about something called glutamine. The easiest way to think about it is like this. It's the cancer cell's favorite high-octane energy drink. They just slurp it down to get all the fuel they need to grow, to divide, and, crucially, to resist treatment. Now, how does the cell actually use that fuel? Well, it needs a special enzyme called glutaminase, or GLS for short. You can picture GLS as the fuel pump. Without this pump, all that glutamine in the tank is totally useless to the cancer cell. So the researchers' idea was beautifully simple. What if we could just switch off that pump? And this leads us to their central strategy. Instead of just using the chemotherapy drug oxaliplatin by itself, they came up with a 1-2 punch. Step 1, block the fuel pump to weaken the cancer cells. Step 2, hit them with the chemo. The whole idea is that a starved cell just can't fight back as well. Of course, having a great idea is one thing, but you've got to have the evidence to back it up. So let's see what happened when they actually put this idea to the test in the lab. The first clue they found didn't even come from a lab experiment. The researchers started by digging into public data from hundreds of actual cancer patients, and they found something really interesting. The gene for one of these fuel pumps, called GLS2, was turned way up in tumors compared to normal tissue. That was a huge signal that, yeah, they were probably on the right track. Okay, so what did they find? Let's look at the results. This chart is all about cell survival. You see, with just the standard drug, oxaliplatin, about 40% of the cells make it. With just the fuel blocker, even more survive, around 70%. But look at that last bar. When you combine them, wow, survival plummets to below 20%. I mean, that's just clear as day proof that this 1-2 punch is way more deadly to the cancer cells than either drug on its own. But killing cells, that's only half the battle. You also have to make sure that any of the survivors can't just regrow and start the whole problem over again. In this survivor test, every one of those blue spots represents a new colony that grew back after treatment. Just look at the difference. Compared to the standard treatment on the left, you can see how few colonies are left after the combination therapy. This means the treatment isn't just killing cells, it's crippling the survivor's ability to bounce back. So next up, they wanted to test the cancer's ability to spread. They did this test that's kind of like a healing race, where they scrape a little gap in a layer of cancer cells and see how fast they can close it. In the top image with just the standard treatment, those cells move in fast to close the gap. But look at the bottom image. With the combination therapy, their movement basically grinds to a halt. Even after 48 hours, that gap is still wide open. That suggests this strategy could be huge for limiting the cancer's ability to metastasize. So the lab results were looking incredibly promising. But the researchers didn't stop there. They brought in some pretty cool modern tech to really refine their strategy and figure out the best path forward. And this is where it gets really clever. They actually used artificial intelligence to analyze the drug combination. They weren't just asking if it worked, but how it could work best. They were hunting for that perfect dosage, the sweet spot.
So here's a simplified look at what that AI came up with. It's called a synergy map. Basically, it's testing all these different dose combinations. Think of it kind of like a heat map, you know? The purple areas are where the drugs don't really help each other much. But the bright yellow, that's the sweet spot. That's where they're working together perfectly. And what's so interesting is that the AI found the best effect wasn't at the highest dose or the lowest dose. Nope. It was a very specific combo right in the middle. And that's absolutely crucial if you want to design smarter, more effective clinical trials down the road. So after all the data, all the lab tests, all the AI modeling, what's the big takeaway? Well, it really boils down to this simple but incredibly powerful conclusion. Blocking the cancer's access to its favorite fuel source makes it way more sensitive to standard chemotherapy. Now, this is still early-stage research, for sure, but the path forward is really clear. The next steps are to test this idea on more types of cancer cells and also in more complex 3D models that act more like a real tumor. They're also going to keep hunting for other metabolic weaknesses, and of course, keep using AI to help design even smarter experiments. And all of this leaves us with a really big final thought. This research seems to point to a real shift in thinking. Maybe, just maybe, instead of always searching for stronger and stronger poisons, the future of cancer therapy is more about outsmarting the disease, finding its weaknesses, cutting off its supply lines, and turning its own biology against it. A future that's a lot less about brute force and a whole lot more about strategy.